Hello students, welcome to today's session on the antioxidant enzymes in preventing the formation of free radicals. I am Dr. Guhapriya here, consultant nutritionist, to present on this topic. The recent development in the field of free radical biology has brought about a medical revolution that has changed the view on human health and diseases. The normal functioning of cells is dependent on a proper balance of pro-oxidants and antioxidants. The former promote the release of oxygen to provide energy needed for cell functioning. In this process, different biochemical reactions takes place which continuously produce various free radicals. If these free radicals are not quenched by antioxidants, they can cause damage to the cells, proteins, DNA and RNA. Cumulative tissue injury, thus caused by free radicals, is now known to underlie the pathogenesis of such diverse conditions as cancer, atherosclerosis, radiation damage and accelerated aging. USDA defined the antioxidants are compounds that protect biological systems against the potentially harmful effects of processes or reactions that can cause excessive oxidation. Moving on to the objectives. The objective involves to explain about the formation of free radicals and their effect on human health, and to understand the function of antioxidants in preventing the formation of free radicals. Free radicals are chemical species with one or more unpaired electrons. Paired electrons spin in opposite directions and their energy is neutralized, while a molecule or radical with an unpaired electron has unbalanced energy and becomes unstable and highly reactive. It can either get oxidized or get reduced. In biological systems, removal or addition of electrons is the most frequent mechanism, which is known as redox reaction. The reduction is the addition of an electron to an acceptor molecule which stores energy. Oxidation is removal of an electron from a molecule to release the energy. During the process of release of energy to perform normal activities of life, electrons are transferred between the molecules, resulting in many reactive oxygen species and non-oxygen free radicals in the body. There are three known free radicals: the superoxide, the hydroxyl and the peroxide. When molecular oxygen accepts one electron at a time, superoxide radical is formed. Superoxide can accept a proton to form a perhydroxy radical, which is also a free radical. Perhydroxy radical has greater solubility in fat solvents and are highly toxic to the cell membranes, which are rich in lipids. In the second step, superoxide takes up another electron, forming peroxide, which combines with hydrogen ions in the cellular fluid to form hydrogen peroxide. Hydrogen peroxide is more stable and hence less toxic than superoxide. But hydrogen peroxide survives longer and can move considerable distance from the site of its origin by entering the circulating body fluids, causing damage along its route. Hydrogen peroxide is not a free radical but a free radical inducer. The toxicity of hydrogen peroxide is further enhanced in cells rich in transition metals, iron and copper, as in RBCs. Iron and copper are present in the respiratory components of all tissues and are sites for splitting hydrogen peroxide into a highly toxic hydroxy radical and hydroxyl ion. This reaction is known as Fenton's reaction. Hydroxy radical is highly toxic but has a short lifespan, cannot diffuse across cell membranes and cannot spread its toxic effects to neighboring tissues. Superoxide, hydrogen peroxide, hydroxyl radicals and singlet oxygen are known as reactive oxygen species. Sources of free radicals and reactive oxygen species. During respiration, very minute quantities of reactive oxygen species are formed in the cells and get inactivated. RBCs with high oxygen and iron content are a major site of reactive oxygen site formation and their inactivation. Respiratory bursts is a process triggered by the appearance of particulate or non-particulate foreign body. Bacteria from contaminated water or food in the gastrointestinal tract or fumes or smoke from tobacco, gaseous or aerosolic pollutants from automobile exhaust, industrial chimneys or dust particles inhaled into the lungs can stimulate the respiratory burst. It is a process by which the WBC, specifically the neutrophils, move into the site, engulf the foreign matter, phagocytize them and digest them in a shift move to prevent their potential damage. During this process, the ROS are formed in excessive quantities together with another toxic radical, the hypochlorite. The process occurs frequently in the mucosal surface of GI tract and the respiratory tract, and the toxins are spread through blood circulation. Detoxification of xenobiotics is yet another avenue for free radical generation in the body. A variety of drugs like antibiotics, antimicrobials, anti-cancerous drugs, analgesics, pesticide residues contaminate the food and the drug sources. The chemicals like benzene and hexane are detoxified in the body by a system known as cytochrome P440 mediated mixed function oxidases, especially in the liver cells. This system is induced by the exposure to toxins and provides greater protection. However, during this process, large quantities of reactive oxygen species are formed, causing damage to the liver cells. Under ischemic condition, the reactive oxygen species are generated in excessive amounts. Auto-oxidation of small molecules like adrenaline, ferredoxin, and even the life-sustaining glucose, when present in high levels, produce the free radicals and endanger the cells and tissues. Several commonly used drugs like paracetamol and dietary constituents like nitrate are metabolized into toxic components and free radicals. Nitrate is present in water and foods. In the acid environment of the stomach, nitrate is reduced to nitrite. The latter is the reactive radical and combines with amines and amides of proteins to form N-nitrose derivatives, which are free radical generators. Nitrosated proteins are abundant in cured and smoked meat, fish, and milk products. Nitrates are present in vegetables like beetroot, celery, and spinach as well, etc. Moving on to disease processes by free radicals and reactive oxygen species. Free radicals and reactive oxygen species abstract hydrogen from membrane phospholipids which contain PUFA, producing reactive free radicals. Oxygen adds on to the site of free radical formation to produce a peroxide in the second step. The fatty acid peroxide now abstracts a hydrogen from the second fatty acid on the membrane, producing a second free radical. This can proceed as a chain reaction with increasing free radical as well as peroxide formation on the cell membrane. Membrane peroxidation leads to fragmentation of the membrane and release of free radicals in the tissues, affecting the structural integrity and permeability. While lipid peroxidation is the first steps on the membrane, proteins on the membrane as well as the cytoplasm are acted upon by the free radicals and reactive oxygen species in several ways like modification, oxidation, hydrolysis, and fragmentation and inactivation. Oxidatively modified protein of the lipoprotein, low density lipoprotein, is known to be the mediator in cholesterol deposition in the blood vessel, causing heart attack and stroke. Free radicals and reactive oxygen species react with the DNA, producing strand breakage, mutations leading to cancer, and also interfere in the regulatory control of growth, differentiation, cell division, and cell death. Moving on to the markers of oxidative stress. There are two assays that are used in markers of oxidative stress. The first of these is the immunoassay of oxidatively modified LDL particles. This is specific to cardiovascular disease risk with dietary and supplemental antioxidant consumption. The second assay is the measurement of the compound isoprostane. It measures the presence of continuously formed free radical compound that is produced by the free radical oxidation of specific polyunsaturated fatty acids. Moving on to the antioxidant defense systems. Antioxidant defense system of the body consists of endogenous and exogenous antioxidants which work together at a molecular level to protect the cell membrane, lipoproteins, DNA and RNA from damaging the effects of free radicals. Exogenous antioxidants are nutrients such as ascorbic acid, tocopherols and beta carotene, and non-nutrients obtained through the intake of different diets. The compounds with established or proposed antioxidant activities in vivo are listed as follows. There are primary antioxidants which includes vitamin E, vitamin C, carotenoid, flavonoid, polyamine, melatonin, estrogen, etc. And secondary oxidants includes copper, glutathione reductase, ascorbate reductase, glucose 6-phosphate, etc. Compounds regarded as a first line of defense, that is the primary antioxidants, or as secondary antioxidants, are listed in the above table. Some are synthesized by cells endogenously and other need to be provided by the diet. For example, a number of enzymes with antioxidant function require trace elements from the diet as cofactors. Cytoplasmic and mitochondrial superoxide dismutase require copper, zinc and manganese to catalyze the removal of superoxide radicals produced by the cell. Hydrogen peroxide is removed by catalase which requires iron and also by cytosolic glutathione peroxidase. This enzyme also removes potentially toxic lipid hydroperoxidase from the cells and is dependent on selenium for optimal function. Non-enzymatic antioxidants. The non-enzymatic antioxidants are small molecular weight compound endogenously produced with examples including glutathione and uric acid. Micronutrients of dietary origin with antioxidant functions include the following. First is the vitamin E, which is an example of polyphenolic antioxidant. Such molecules readily donate the hydrogen from the hydroxyl group on the ring structure to free radicals, which then become unreactive. Vitamin E is particularly effective in preventing the lipid peroxidation, which is a series of chemical reactions involving oxidative deterioration of PUFA. On donating the hydrogen, vitamin E becomes relatively unreactive free radical, which is unable to attach the adjacent fatty acids because the unpaired electron on the oxygen atom becomes delocalized into the aromatic ring structure. Lipid peroxidation may cause disruptions of cell structure and function and thus plays an important role in the etiology of many diseases. Although vitamin E is primarily located in cells and organelle membranes where it can exert its maximum protective effect, its concentration may only be 1 molecule for every 2000 phospholipid molecules. This suggests that it is rapidly regenerated possible by vitamin C. Next is vitamin C, which is the L-ascorbic acid, is one of the most important water-soluble antioxidant in cells. It is synthesized in the livers by most animals but not by men who depends on dietary sources for adequate supply. The ascorbic acid is a strong reducing agent and serves as an antioxidant and as a cofactor in hydroxylation reactions as well. Vitamin C effectively scavenges a range of reactive oxygen species, peroxyl radicals and singlet oxygen, and can also chelate the trace elements such as iron and copper, thus inhibiting the catalytic decomposition of hydroperoxides to potentially damaging the products. Moving on to the beta carotene. Beta carotene is effective at scavenging the singlet oxygen. It is also an effective antioxidant at low partial pressure of oxygen under conditions where singlet oxygen is not formed and therefore may be important at the physiological oxygen tension in tissues. There are many other carotenoids in foods that are also found in the tissues. Their nutritional and biochemical significance is poorly understood but may be highly specific as some tissues accumulate particular carotenoids. For example, the lycopene is a major carotenoid in the testes and macula of the retina, contains high concentration of zeaxanthin and lutein. Moving on to ubiquinone is a lipophilic quinine which functions as an electron carrier in the mitochondrial electron transport chain of the cell. However, it has also the antioxidant properties in vitro. Ubiquinone is synthesized in the body from precursors of cholesterol. For this reason, it is not classed as a vitamin. However, the ability to synthesize ubiquinone decreases with age and there may be an increasing dependence on food to supply the nutrients as well. Moving on to food polyphenols such as the flavonoids, chalcones, cinnamic acid, coumarins and anthocyanins are ubiquitous dietary components. Originally regarded as being nutritionally inert, there is now increasing interest in their antioxidant properties. Indeed, low intakes of flavonoids such as quercetin, myrcetin and camphorol have been associated with increased risk of coronary heart disease. Moving on to the enzymatic antioxidants. An interacting network of antioxidant enzymes protect the cell against oxidative stress. The superoxide released during oxidative phosphorylation is converted to hydrogen peroxide and then reduced to water. This pathway is a result of various enzymes with superoxide dismutase catalyzing the first step and then catalases and various peroxides removing the hydrogen peroxide. Moving on to superoxide dismutase, which are the group of related enzymes that catalyze the breakdown of the superoxide anion into oxygen and hydrogen peroxide. These enzymes are in almost all aerobic cells and in extracellular fluids. The three major family of superoxide dismutase, depending on the metal cofactor, the copper or zinc, iron and manganese, and finally the nickel. In higher plants, superoxide dismutase isozymes have been localized in different cell compartments. Manganese superoxide dismutase in mitochondria and peroxisomes. Iron SOD in chloroplasts but also been detected in peroxisomes, and copper zinc in cytosol, chloroplast, peroxisomes and apoplast. Catalase is a common enzyme present in all living organisms which are exposed to oxygen, where it functions to catalyze the decomposition of hydrogen peroxide to water and oxygen. The byproduct hydrogen peroxide is harmful and must be immediately converted into other less dangerous forms. Hence, catalase catalyze the decomposition of hydrogen peroxide into less reactive gaseous oxygen and water molecules. This is found in high concentration in liver cells. Glutathione systems include glutathione, glutathione peroxidase, glutathione reductase, glutathione transferases. This is found in animals, plants and microorganisms. Glutathione peroxidase catalyzes the breakdown of hydrogen peroxide and organic hyperperoxide in animals, as it contains 4-selenium as a cofactor. Glutathione peroxidase 1 is the most abundant and is very efficient scavenger of hydrogen peroxide, while glutathione peroxidase 4 is the most active with lipid hyperperoxidase. The glutathione S transferases show high activity with lipid peroxide. These enzymes are at particularly high levels in the liver and also serve in detoxification metabolism. The endogenous antioxidants are detoxifying enzymes which are primarily physiological in origin. In phase 1, drug metabolizing enzymes such as mixed function oxidases plays a major role in detoxifying the toxic chemicals. In phase 2, many non-enzymatic organic molecules such as glucuronic acid, glutathione, glycine bind to the functional groups of the toxic chemicals or to the resultant phase 1 metabolic products and form polar compounds in order to facilitate their excretion from the body. Different antioxidant enzymes are involved in phase 2 metabolism such as glutathione S transferase, NADPH, quinine reductase, UDP glucuronil transferase and epoxide reductase. The activity of these enzymes is enhanced by several non-nutrient phytochemicals that are present in different foods which are useful in detoxifying the toxins. In this phase, the balance between glutathione S transferase and quinine reductase determines the toxicity of chemical carcinogens. These enzymes require certain trace elements such as selenium, zinc, copper and iron can be obtained through dietary sources. Major antioxidant mechanism involved in fighting the free radical damage, which includes the interaction of ascorbic acid and glutathione with oxidants and oxidizing agent. Second, scavenging of free radicals and singlet oxygen by vitamin E, ascorbic acid, beta carotene and superoxide dismutase. And reduction of hydroperoxides by glutathione peroxidase and catalyze enzymes. And binding of transition metal by various chelators and repair of cellular damage by various metabolic activities. Moving on to combating the free radicals and reactive oxygen species. Potentially injurious effects of free radicals and reactive oxygen species are prevented by a well-organized antioxidants defense system. The first line of defense is prevention. The multi-step reduction of oxygen to water in the mitochondria is carried out by the cytochromes and other metalloenzymes held together so that the intermediates like superoxide are not released into the cell. Similarly, ions of transition metals like iron and copper, which can trigger the reactive oxygen species formation, are bound or sequestered by protein-like transferrin and ceruloplasmin to keep them out. The second line of defense is provided by the antioxidant enzyme present in two cells. They speedily attack the reactive oxygen species and inactivate them. Superoxide dismutase reacts with superoxide to give less toxic hydrogen peroxide as follows. Hydrogen peroxide is inactivated by two distinct enzymes. The catalase acts on the molecule, splits it into water and oxygen as follows. And a second enzyme, glutathione peroxidase, detoxifies hydrogen peroxide using a donor antioxidant such as glutathione. GSH can exist in two interconvertible forms as follows. It can give hydrogen to combat oxidative stress. It can also take up hydrogen and can be considered as a storage form of antioxidant. While combating free radicals and reactive oxygen species, glutathione gets inactivated to the oxidized form but can be regenerated by the cellular metabolism using another enzyme, glutathione reductase. The hydrogen donor is a nucleotide carrying the vitamin niacin. The enzyme contains selenium. The dietary deficiency of selenium and niacin also lead to antioxidant deficiency. The enzyme superoxide dismutase contains copper, zinc and manganese. Dietary deficiencies of these minerals also lead to antioxidant insufficiency. Riboflavin is needed for maintaining the niacin containing nucleotide participating step in GSH regeneration, making riboflavin also as an antioxidant. The third line of defense is the damage control provided by the free radical scavengers. They react rapidly with the free radicals, inactivate them and arrest them chain of propagation. In the process, they are inactivated into less toxic radicals. The scavenger active on the cell membrane is vitamin E. The inactive form of tocopherol on the cell membrane is regenerated on the cytoplasmic side with vitamin C as a hydrogen donor or free radical scavenger, rendering the vitamin oxidized and inactive. This in turn is reduced and regenerated by glutathione. The resultant oxidized glutathione is reduced to provide a continuously supply of GSH by a cellular enzyme glutathione reductase, which in turn needs the coenzyme of the vitamin niacin, NAD and NADP. These cofactors are provided by glucose metabolism with the help of coenzymes of riboflavin, and these vitamins also share the work of free radical scavenging. The final defensive is by the repair of damaged molecules and structures by accelerated removal of damaged molecules. For example, the LPO products on the membrane is removed by specific phospholipases which cleave the damaged fatty acyl chain, which is followed by replacement with fresh fatty acid. Damaged protein are digested by proteases and damaged part of DNA are cleaved and repaired. The repair mechanism is limited and is not exhaustive, leading to diseases and disorders. To summarize, the release of energy to perform normal activities of life result in many reactive oxygen species and non-oxygen free radicals in the body. Antioxidant defense system of the body, endogenous and exogenous antioxidants work together at the molecular level to protect the damaging effects of free radicals. Hope the session on the free radicals and the prevention is useful. Thank you.